I'm going to invite Elliot, Andrew, and Anna Marie to the stage, and we're going to talk a little bit more about IP Salvage. Oof. Cool. Any order. Cool, thank you. So yeah, we're gonna talk about failure, but not in a depressing way, in a hopeful way, like what can rise from the ashes of failure. Because famously, 90% of startups fail. And when that happens, especially in biotech, those assets normally vanish, leading to systematic amnesia and repetitive multi-million-dollar learning curves, which I think is a real waste of scientific knowledge and progress. So a year ago, or over a year ago actually, when a cultivated beef company named Sci-Fi Foods failed and went bankrupt, they put their assets up for auction. And then GFI and Tufts teamed up to acquire their cell lines and media formulation so that others could sort of pick up where Sci-Fi left off. And specifically, they deposited those cell lines into an open access cell bank here at Tufts, which is a project that's still unfolding and which we'll talk about today, and which I'm sort of working on scaling as a fellow at Renaissance Philanthropy, trying to think about whether IP salvage can be a sort of reusable model for innovation that can be deployed across domains. So not just in alternative proteins, but other deep tech topics like carbon removal. So we have a really great collection of people here today. So we have Andrew and Elliot, who really spearheaded the Sci-Fi acquisition, and then Anna Marie, who provides a sort of zoomed out perspective from her time at Ginkgo Bioworks, where she led, I think, like a dozen acquisitions of other failing biotech companies, and is now at a stealth startup thinking about how to sort of capture tacit knowledge and other things. So we'll do a brief round of introductions and then get into it. Yeah, do you want to start?
Sure. And your intro is actually apropos because something else I do that is not on my normal bio is I teach a class on failure at HBS. So failure has been an obsession of mine, partly just exposure therapy, I think, because this industry is so hard, but it's a topic I love. As Meera mentioned, I was at Ginkgo for 5 years. I led a number of functions there, but including strategic transactions. So we acquired about 20 companies, most of which were in this category. Great science, but the money ran out. And rather than letting that great science evaporate into the ether, could Ginkgo be a home for that science and often those scientists to make sure that that platform technology wasn't lost? So this is a topic I care deeply about. When I left Ginkgo, I was obsessed with a couple things. One was, why is the business model of science so darn hard? Why don't we have platform technologies in the sciences in the same way that the tech industry does? I was also leading AI at Ginkgo, and we had this issue in AI where we fundamentally have no data provenance in this industry. We have a hypothesis, we run an experiment, we get some results, we assume that those results have something to do with the hypothesis, yet we have zero visibility into the experiment that was actually done and whether it was done correctly, whether the weather was different today, whether somebody made a mistake, whether a reagent was expired. Very rarely do we really have that metadata. And so when we look at AI and training data at scale, you often are finding batch effect and site-to-site variability is more prevalent in the data than actual biological function. And that inspired the formation of Transfyr, which is the company that I founded earlier last year and am leading now, which really brings those two topics together. It's recognizing that often the challenge for the business model of science is our inability to transfer and distribute the tacit knowledge that actually makes science work in somebody else's hands. And if you can't distribute your product, you probably don't have a business. And so this is, again, a topic that we care deeply about, and trying to enable the future innovators of the world to be able to distribute their technology and capture the value of the science they create, rather than facing the sort of lottery ticket of biotech right now.
No. Yeah. Hi, everybody. My name's Andrew Stout. I'm an assistant professor here at Tufts in the biomedical engineering department. I started that role this past September, so still fresh, but I've been at Tufts for a long time. I did my PhD here under David Kaplan, focused mostly on cell line and media development. And then after finishing grad school, myself, Natalie, and John started the Cell Ag Commercialization Lab here at Tufts and associated spin-out Deco Labs. And one of the projects that we were really interested in at the Cell Ag Commercialization Lab was the idea of this open access cell bank. And so, yeah, I'm really happy to talk more about it today.
Thanks, Andrew. I'm Elliot Swartz. I'm a senior principal scientist specializing in cultivated meat at the Good Food Institute. My background's mostly in stem cell biology, and I've been at GFI for the last 7.5 years, working on various topics related to cultivated meat. So that's, environmental impact, food safety, but really spend most of my time thinking about cost and cell culture media, and have been involved in some other special projects like this IP acquisition that we'll chat a little bit more about.
Great. Yeah, let's start there. So maybe could you fill us all in, or fill other people in, I was there, on the background, like how did the sci-fi acquisition come to be? Was it like a whirlwind thing? Was it in the works for a while? How did you decide what to and what not to acquire? And yeah, Andrew, please chime in too, or fill in the blanks.
Sure, yeah, I can start. So, myself and others at GFI, we obviously are talking to companies on a fairly regular basis, so we have a decent pulse about what's going on. And we had known for a bit of time from Josh March, who was the CEO, that they were in some financial troubles and they were looking for ways out, either to sell the business, get acquired, sell their facility, et cetera. Eventually, one day, I got a text from Ilya, our then CEO, who said, hey, can you sign this NDA? Sci-Fi is going to have an auction for their assets. Can we take a look? We're interested in potentially acquiring some of these things. And we had been in contact with Andrew and other folks at Tufts, around this auction. And so we did some, diligence into what assets were available. They actually had, dozens, if not hundreds of cell lines. But, the Sci-Fi team was actually very helpful in informing us, hey, these are the ones that we're planning to commercialize. These were the best performing ones. And so that helped sort of narrow down the list. We couldn't logistically really be acquiring and shipping hundreds of different cell lines and thousands and tens of thousands of different vials probably. So we needed to limit the ones that we thought would be most valuable for the field as research tools. The media formulations as well are obviously a sort of a package deal. I think without the media formulation, the value of the cells really plummets. And so eventually, GFI sort of put together a bid. I can't state the exact value, but it could say that, it's probably less than what you might be thinking. And so that, made us pretty confident that, taking a look at what Sci-Fi had developed and the value that we needed to put in, that we could provide a lot of long-term value for the field, get a really good return on investment, so to speak, for the entire, ecosystem. If we could execute this, that bid was accepted. We got the vials shipped over to Andrew. So I don't know if you want to fill in any other blanks, but we've been sort of working in the background on wrapping up the sort of last legal aspects, and hopefully we'll be able to distribute these cells soon.
I guess I'll just briefly fill in about sort of what the cells are, because I think it's an important part of the story of sort of the conversations that we had about whether it was worth putting in a bid. And so the cell lines, it's 8 cell lines, 1 adherent bovine muscle cell line, and then 7 bovine fibroblast lines, many of which are, they grow in single cell suspension. And I think what was so exciting to me, and I think to Elliot as well, is that historically, there's been a real mismatch between the sort of formats of cell culture that academia has been using and the formats of cell culture that a lot of industry has moved towards. So industry, many of the big players have moved towards suspension cell culture because that's the type of cell culture that's most amenable to being grown in large bioreactors. And academia had really been focused on really small scale adherent cell culture. And I think just the real value that struck me about these cell lines was it could just open up that new mode of cell culture for industry and for academia, and could kind of bridge that mismatch and bridge that gap in a way that I think is really exciting. And I think both systems are important and relevant, but there was one that had previously been missing for a lot of folks who weren't at the companies that had already developed single cell suspension lines. Yeah.
Cool. And so now the cells are at Tufts in this open access cell bank. I feel like the idea of a cell bank has been floating around in cellular agriculture for a really long time, like going back to sort of our days at New Harvest, so 7-ish years ago. What do you think took so long to get a cell bank up and running, and how has the Sci-Fi acquisition sort of like changed your thinking regarding how a cell bank should be structured in terms for distribution, things like that?
Yeah, I think so. I think what took so long, I don't know why it took so long. It shouldn't have taken so long. I think it's a lot of little things. So I think in some ways, this cell bank is extremely exciting to me, and I also think it's sort of in some ways not sexy to like the big pillars of the type of people working on this work. So academia is mostly interested in developing new cell lines with new technologies, exploring new questions, asking basic biology questions, and industry is really interested in creating kind of the best cell line that they will carry through to production. And what I think the sort of cell bank effort is aimed at producing sort of baseline cells on which all other efforts can be built up. But it's not going to necessarily get you, if I produce another cell line using a similar technology that people have already published on, and I kind of know it's going to work. It's not sort of the sexiest paper that an academic could publish. And a company that makes sort of a baseline cell line has absolutely no interest in giving that resource to other folks to use. They want to protect that cell line. And so I think on the industry side of things, and sort of, I guess, stepping back in time a little bit, I think at the start of the existence of this field, when it was new, there wasn't a really clear sense of what the valuable intellectual property that a company had was, and so you protected all of the intellectual property that you had, and there were lots of patents filed on things that probably had a lot of overlap with other patents or papers that had been published, and there was sort of a real sense of, well, we're just going to protect the whole stack like crazy, and I think that just sort of has changed a little. I think there's sort of a nuanced understanding now that the intellectual property that's going to set one company aside from another is not that they have an immortalized bovine cell line. It's their process behind that cell line. It's the reactors that they use. It's further developments and optimizations of those cell lines. So I think there's a little bit of that bottom layer maybe doesn't need to be sort of as unique and protected across different players. And then I think what the Sci-Fi story really did for me in terms of changing my thinking about the cell bank is just kind of like opening the idea to the realization that, if when we're talking about this cell line or this cell bank, we're saying, hey, all these companies are making these redundant cell lines. Let's make some cell lines that everybody can use. So let's stop the redundancy in a year's time when we make this cell line. And the Sci-Fi acquisition makes it possible to stop the redundancy now. We don't need to do a redundant effort of making suspension bovine fibroblasts. We can just get them from Sci-Fi or potentially others from future companies as well. So it just increases the efficiency, I guess.
Yeah. So when we submitted the bid for Sci-Fi, I think like the other option was that like a different company could have acquired the cell lines, and it would have been like absorbed into some other like private entity, which is kind of what you did with the Ginkgo acquisitions. So I'm curious how you decided what companies to acquire, and was that normally like through the bankruptcy process or prior to that? And then what normally happened once you did acquire their IP or their assets? Like, did it ever sit on the shelf, and then it could have been more counterfactually impactful if it had been salvaged in the way that Andrew and Elliot did with Sci-Fi?
Yeah. So, I mean, we did a number of different acquisitions. They all had their own unique flavor. I would say it was a combination of what we would call proactive. So that was identifying key gaps in our platform at Ginkgo, and then identifying the companies, the younger companies that would potentially be actionable, that had that type of technology, and then building the relationship and being there when they were open to a transaction. So those would have been more proactive, more expensive transactions. We were also opportunistic. So when assets were in distress, we had a good pipeline to make sure, and like, I mean, to the point where we explored things like putting a one-page term sheet up on our website where somebody could just assign IP to us if they were going out of business. You know, we were trying to make it that easy to do it for a whole bunch of reasons. It is never actually that easy, so we didn't do that. But we became known as a place you would call if you were in that position, and so we got a lot of deal flow, which allowed us to sort of take our pick of what assets we thought were valuable. Typically, these assets still had a core team around them, so we were able to benefit from the expertise of those teams. And one of the things that was hardest for us to manage, and partly the inspiration behind Transfyr, was that each of these acquisitions would typically take upwards of a year before Ginkgo's team could reliably reproduce the technology of the acquired company in other sites. And so that was the actual burden to doing the acquisitions, was that tech transfer cost, the training across sites from the original team to sort of our core facilities that would be running that technology or scaling that technology up to our partners. You know, I can't think of a technology that truly just sat on the shelf. One of the advantages that Ginkgo had were relationships with lots of enterprise customers. And so our sort of secret advantage in all of these processes were we could, you know, we had a 90-person sales team, and most of the companies that we were acquiring had a one-person sales team, and that person was also the CEO who was trying to not let the company run out of money. And so we could very often, and we had all these other assets we could surround this acquired asset with to offer a much more complete package to an acquirer. And so we were able to acquire companies for $1 million and turn it into a $40 million enterprise contract, 6 months later. And so that was the real thesis behind a lot of those tools. But I mean, to your point, Meera, like you're still at the whim of the priorities of a corporate acquirer. And one of the things that when I was leading that team, was very careful about was trying to not do what we saw a lot of other corporate acquirers doing. And so most often what happened, even for successful companies, they'd get acquired predominantly by pharma. Pharma would be acquiring them for the asset, and then the core technology that had led to that asset would just sit on the shelf. It would never be used by that pharma acquirer. They would not let it out. And so most corporate acquisitions do exactly what you were worried about, which is you take an asset just to take it off the field for other potential competitors and not to actually do anything with. And so this idea of creating open access platforms for platform technologies is one that's really interesting. The bottleneck that I worry about and that I hope that we can help with is, okay, great, now a whole bunch of people want to access that cell line and that media formulation. How do you make sure they are successful with it and you're not dealing with a, human to human 6 to 12 month long tech transfer process for every single partnership? How do we make that distribution efficient?
Yeah, maybe we can pick up from there. So like when we got the sci-fi cells, and Andrew, maybe you can talk about this, like just relying on sort of the generosity of former members of the sci-fi team that were just voluntarily answering questions. So like if we were to continue doing IP salvage, like how would you improve the sort of like tacit knowledge transfer as opposed to like just getting the cells?
Yeah. So I guess it's a good opportunity to also just say that there was so much generosity sort of across the board involved in this. I think GFI sort of stepping up for this acquisition was extremely awesome. On the Tufts side, there have been a number of donors who've sort of supported the early cell expansion work, the Coley Carlson Foundation, the Astera Institute. There's been a lot of support that's been extremely helpful, and that I think they wouldn't have sat on a shelf, but they would have sat in a liquid nitrogen tank. And then sort of on the sci-fi food side of things, I mean, it makes sense, but the founders of the company and scientific ex-employees of the company were really generous with their time to hop on calls and just talk us through troubleshooting, talk us through the cell culture process, the techniques, all that sort of stuff. All those little tricky bits that are
magic hands.
Yeah, exactly, the magic hands. And So I think we've kind of talked about this a little bit, Meera, but if there was sort of a way to incentivize that, yeah, magic hand transfer, or give ex-employees, I don't know, little internships or these kind of transfer opportunities so they could come sort of help you figure out the cell culture, help teach folks at the cell bank in this example how to use the cells and the systems effectively, then I think it'd be really great, because it was great that these folks were willing to help us, and yet you always feel sort of bad sending an email that's like, hey, can I steal another hour of your time for free from the company that you got laid off from? There's probably some trauma associated with it on their end. Yeah, so I think if there was a way to kind of give back to them for giving back, because I think all these people want to, right? They care a lot about this intellectual property. It was like a lot of their time and a lot of their mental energy, and they're proud of these cells. So if there's just a way to kind of ease that, it'd be really great.
Anna Marie, how would you recommend we do that? Like, would it be deploying your sensors prior to the?
Let me tell you about Transfyr. Yeah. So this is what our company does. So we're fully focused on how do you elucidate and capture the tacit knowledge, the magic hands, the taste, the muscle memory, the intuition that is so critical for getting science to actually work reliably. And one of the things we've been talking about, with Meera is in these situations, could you almost time capsule what the scientists are doing before a company winds down, so that whether it takes a month or years for that time capsule to be opened and somebody wanting to now reproduce and scale up that technology, you have as much rich information as you possibly could. So what we do, we sort of describe it like Waymo for science. The way we got self-driving cars was not just uploading a bunch of maps to an AI model. Same thing, we're not going to get autonomous science just by uploading a bunch of protocols to an AI model. The way we got self-driving cars was we put cameras and LiDAR on cars, and we taught models how to understand the world around the car and how a driver navigated through that world. And so we believe that if you want to get autonomous science, if you want to get reproducible science, if you want to get transferable science, you need to do the same thing. You need to sensorize the lab, understand the lab environment, and understand how scientists navigate through an experiment. So that's what we do. We put cameras, audio recording, environmental sensors, supply chain monitoring in the lab. We very much partner with the scientists who are actually doing the work. So we want them to love the product. And so whether it's simple things like, hey, we'll write your lab notebook for you, or you can get instant replay of what you just did. So when you skip a well, you know you skipped a well and you're not just relying on your memory. But there's this sort of higher level goal around understanding what causes an experiment to work, what causes an experiment to fail, where does variability come into this process? Can we start figuring out what are the magic touches? And quite often, it's not about how well somebody follows a protocol. Often the protocols are wrong, they are sub-optimized, they are underspecified, and so can we bring that color in where it matters and help people understand which steps of a protocol you can be kind of sloppy with and which things really, really, really matter to help that taste be more transferable to somebody who might be picking it up cold and trying to get it to work for the first time.
Going back to the sci-fi piece a little bit, so it took a year for us to make the announcement, but once we did, there was a huge influx of interest. Maybe Elliot, could you provide some updated numbers from the interest form of people that have requested access to the cells and how that breaks down by academics versus industry folks?
Yeah, so we made an announcement. We have a waitlist form at the moment while we get the last final pieces settled before distribution, and there's been 40 requests thus far. About 22 of those are academic labs around the world, and 18 are different companies around the world. So that, I think, is pretty good. It's a good signal to us that people also see the value in these cells and being able to just sort of level up and jump in at a starting point that allows you to skip probably years of R&D and millions of dollars. So a lot of people are interested in that, and I don't blame them. And so yeah, our goal will be to get them in the hands of as many hands as possible. And also, I think what Anna Maria mentioned earlier with respect to deal flow, once you start doing this, now that we're in this environment where there are companies, going under and others that are in, financial worry or looking for options, and everyone's really mission driven in this industry they don't want to see their work go to waste and be shelved. They like to see it lived on. Now that we have this news out and we're starting to popularize this concept of IP salvage, I think people are starting to come to us with their own ideas and starting to get more conversations and this quote unquote deal flow coming in, which I think is really great because I think in the long run, this can be a huge boon, as Jan was mentioning, into supporting the R&D ecosystem, not only in academia, but also in industry as well.
And do you see salvage opportunities outside of cell lines specifically?
Yeah, so I think for us at GFI, I mean, we've had some conversations internally about, what would we prioritize. I think cells and media seem to come at the top of the list. These are kind of like, as Andrew was mentioning, maybe the most important like pre-competitive assets that could exist where, you get to this point that, you're sort of ready to start to have something that can be grown in relevant bioreactor settings and bioprocess settings and these sorts of things. And then, you can always build on top of that. But getting to that starting point alone takes years and millions of dollars, et cetera. And so cells and media are really priority, but, also things like data sets could be of interest, especially with people wanting to apply AI and machine learning. We've had discussions about, what types of sensory data or bioprocess data could be available. Of course, there's a lot of problems with that too. Different companies might call a batch something else. There's different ontologies, the order of the data structure, et cetera. So those are questions on how high utility acquiring data sets could be, but it's something that maybe we'll see happen eventually. And then there's also equipment and facilities themselves that could potentially provide value in different ways. Facilities could be transformed into co-manufacturing sites for the shared facility and infrastructure, for instance. I don't know if that's something that GFI would particularly lead, but obviously there's other people, including yourself, that are taking a look at these sorts of opportunities and coming up with the best way that seems to be of the broadest benefit.
And why put the cells at Tufts versus like ATCC or a more established repository?
Should I?
Yeah, go for it.
I guess it was your decision. So I think one of the things that I'm really excited about and really grateful to Tufts for, and to the Tech Transfer Office in particular for, has been their sort of willingness and enthusiasm about trying to build like what's really an open access model for these cells, and it all involves kind of buying into the idea that this intellectual property at this cell bank is really just the starting point for future intellectual property development and for commercialization. And therefore, not only is it nice to not protect it too much, but as soon as you protect it too much, it ceases to have value or interest for others. And so what I really mean by that is that the Tufts cell bank, we're basically able to do it with a royalty-free distribution. And so the vials will be available just for the cost of sort of expanding them and shipping them, just sort of like the internal expansion and banking aspects. But even for commercialization, there won't be royalties charged. It'll just be a one-time transfer. It's built on, we didn't invent this model. There's a cell bank called Open Pichia out of Belgium that does this for Pichia pastoris. And I think it's just a really cool model because I think for this sort of, for these cell lines that are the baseline starting point, as soon as a company who would potentially acquire those cell lines looks down the line and says, okay, well, if in 10 years I owe some percentage of my profits to these folks who gave me this pretty, at the end of the day, this kind of starter cell line, then I'm just going to make the cell line myself. And I think that's why there's been so much redundancy, is that you don't want to be beholden to anybody for something that's so upstream in your whole sort of IP development and your whole sort of production pipeline. And so the fact that Tufts has been sort of willing to embrace those terms and is game for that sort of royalty-free distribution model, I think is exciting, unique, very cool, and not something when we initially kind of chatted with folks at ATCC or others, that wasn't like the model that they operate under for commercial use of something.
Yeah, I'll just add that there's probably no one in the world that has thought more about open cell line development than this guy right here. As someone that's read a lot of proposals from different scientists in cell line development and that sort of thing, Andrew and the team here have thought quite a bit about how to develop cells that would be of the highest utility and also the highest value and openness and freedom to operate in the field. So that also gave us pretty high confidence, in addition to like the physical infrastructure that's going to be here to partner with Tufts and hopefully keep that going.
I'm going to start taking Slido questions, of which there are many. Maybe we'll start with what are the downsides of a nonprofit providing assets for free in a way that discourages private companies from entering and building a sustainable market?
Yeah, so there's obviously B2B companies in the sector that are developing cells with the intention to license them to industry, and that's how they would run their business. So there is there's some degree of overlap and interference here, I guess you could argue. It should clarify these cells are not going to be for free. So there will be a price attached to it, and that price will go, as Andrew mentioned, to maintain the operations at the cell bank. In addition, with the sci-fi situation, there's also third-party licenses that have to be considered. Those cells were edited with CRISPR, and so these sorts of considerations are going to be case by case on a cell line development basis. But that's why there are technologies that enable you to have freedom to operate and still engineer cells, and so those are probably good to use if you're developing cell lines just in general. But I think from an ecosystem perspective, the more research that's done using high quality cells that are well characterized, the better the whole field goes. I mean, if you're developing cell lines for the industry, you want the industry to exist in the first place and be successful with products on the market. And the way that we get there faster is by having better research, more research, et cetera, all around the world. Additionally, the cells that you might specialize in creating, if you're a company, could be very unique and niche to different customers that you might have in mind or partnerships already in place. And that's sort of different than what we're doing here, which is just to get cells that can perform pretty well as a starting point. And so I think there's differentiation opportunities that are quite available for you to still work.
Yeah, can I piggyback off that? Yeah, I mean, if these cells are the best cells, then we messed up, right? Like, these are supposed to be cells that other folks build on and build better cells. And what I would like to see is I'd like to see banked cells get better and better and better year after year. And then companies that are working to produce products or to sell, really, really specified cells also get better and better and better and better every year. And one of the examples that we talk about a lot is like on the microbial production side, right? Anybody can buy a vial of E. coli, and no huge company is going to buy DH5 alpha E. coli and then produce their products in that. They're going to have optimized the hell out of those cells, but that doesn't mean that oh no, we shouldn't sell cheap DH5 alpha sort of basic E. coli as research tools, as starting points, and all of that, because it kind of is anti-competitive with folks who are making better strains or who are developing additional strains. I think that when we think about cell lines for cultivated meat production, we tend to sort of lump it all into a bucket, maybe with some differentiation that we think of. We think of primary cells, we think of immortalized cells, we think of maybe adherent and suspension cells. But I think in another 10 years, there's going to be such a huge range when you look at sort of the efficiency, effectiveness of a primary cell isolated straight out of a cow and sort of the most optimized cell lines that folks are using in industry. And we sort of do ourselves a disservice by sort of condensing the conversation around kind of putting all of that into a single bucket.
I might just add a little macro point. Look at the tech industry. So much of the tech industry is built on open source software. It is why that industry has been able to grow as fast and as well as it has. There's an enormous culture of people developing stuff and then just putting it on the internet so that other folks can build on top of it. We just got our very first $1 trillion company in the biotech sector last year. And we had, what, 5 tech companies that were over, $5 trillion tech companies. Like, why is it that the tech industry, which is arguably not helping society right now, like worth so much more than this critical infrastructure of human health, human food, environment? Like, it's absolutely nuts. And so I really think there's this like cultural element and fear around like, oh my God, there's so much, like I need to protect my IP, I need to lock everything up, I need to hoard it. Candidly, because it's been so hard to monetize it and to distribute it, that if we change that technology to be able to change that culture, there's an opportunity for this industry to grow and advance so much more quickly than it has to date. And I'd love to see that happen.
Can the banked, I'm going to expand on this question a little bit, but can the banked cells from sci-fi get approved by the FDA? And I guess to expand on this, like what would be unlocked if there was an IP salvage of like an FDA approved company, or if we salvaged like FDA, USDA approved cells?
Well, I'll take the direct question is like, yes, in theory, they can be approved by the FDA. I mean, Sci-Fi had submitted a dossier to the FDA using one of the cells that we have and that we'll be distributing and prioritizing distribution of. Obviously, there's other things related to, getting approval by FDA, and your process, et cetera. But assuming all those things are in line, in theory, yes. Then that is the goal, is to get these in as hands as many possible and as many use cases as possible, and that means if downstream food production. And so the other question was,
Like,
half.
what would be different if there if you like IP salvaged a
so
FDA
if you were to acquire assets that had already been approved, I feel like I just heard the answer to this question at some point recently, but I think it's also going to vary on a case-by-case basis, depending on exactly how much you're taking, whether it's exactly as is. I mean, it would be hard to, let's say, manufacture that product in the same site, if it's moving somewhere else. And so I think it's not really a one-to-one, like, this is approved, therefore it's instantly stays approved. I don't know, Eric, if someone else might be able to answer that better than I would.
Oh, you responded. What'd you say? Possibly, if the accompanying production process is cleared, FDA could potentially clear a cell line for safe use via safe bacterial strains, but no one has attempted. Generally, FDA wants to approve cells in context, so within a production system. Thanks, Eric.
Thank you. I think I said some of that. Sounded similar.
Does Transfyr have a miniaturized version of their tech? For example, an AI cam or mic for your undergrad, as opposed to needing full-fledged lab recording infrastructure? And like, can people in the room can they come to you? Can they use Transfyr?
You should definitely email us. We are building one that is appropriate for more academic use. Our focus thus far has been on enterprise customers, but we are exactly right now in the process of effectively downscoping it exactly like this. So that could be like literally something you can just run off of your iPhone or an iPad in the lab, to something like what was described there, which is a single camera, egocentric or stationary, that could be integrated with the system. So not yet, but very, very soon. So please shoot a note if you want to be among the beta testers of that.
And then how do we incentivize other companies to contribute to the cell bank? I think you talked about this earlier with like having deal flow, now that people know that you're in the salvage game, Elliot. But I'm curious if this is something like, what do you think? How do you incentivize people to do this?
Yeah, it's hard because incentives generally imply like you're getting something back, and in this case, I mean, what you're really getting is value that you created that you don't want to see go to waste, and so it's not necessarily. I mean, there can be some monetary value tied to these sorts of things, but it's certainly not enough to equivalent to like a fundraising round or anything like that. So the incentive has to be different than money speaking. But as I mentioned, I think there's a lot of mission-driven people and founders at these companies. And I think one of the interesting things from my perspective that I think is a big opportunity is that we know companies have spent a lot on cell line development. They have cells from various different species in their tanks. A lot of them aren't, going to be used. You really have to focus on one thing, get it to market. I mean, that alone takes a lot of effort. And so, what if you can just kind of give those cells away? And in the meantime, if people start using those cells, they're basically doing R&D for you. And so that's how I would kind of frame it in thinking about, what assets are on your shelf today that can benefit the ecosystem that you could view as free R&D for you that you can eventually build on in the future, and maybe get a small amount of money as well. I don't know if you have different thoughts, Andrew.
No, I think that's exactly my thoughts as well.
Yeah, we thought a lot about this at Ginkgo actually, and one of the hardest parts about this, to the point on like money doesn't actually work, is that often money actually makes the deal more expensive in a way. Like they have to keep the company alive, they have to get agreement from their funders, from their debt holders, also. And so like the promise of like maybe you'll get a few tens or hundreds of thousands of dollars a few years from now is actually like negative value. And so it really, in our experience, it really did come down to you want to see your work live on and you want to have an impact, and people are mission driven in this industry, and so they want to see something just not die. Although I will say, I think one of the most interesting problems in this field is like the attribution question. If you have a product that emerges 5 years from now, it's presumably a mishmash of dozens of different technologies that went into it. And so how do you provide the right attribution to like how much of that value was created by this piece of IP, this piece of IP, this piece of IP, this piece of IP? Because if you can do that in a more systematic way, you have opportunities to do like much more flexible deal structures. Because right now what happens is like, well, I need to pay 5% to such and such for my CRISPR license, and I need to pay X% to such. Like there's no fluidity for like, but CRISPR is not that important in this particular thing. It's like this little tiny piece. And so if there were a way to more flexibly figure out how much value something created in an end product, that might be able to open up a lot of really interesting opportunities for financial incentives and financial markets, and that plugging into some of these like nouveau funding things like Research Hub and all sorts of creative ways to fund the science could be an interesting trend to watch.
Now that the cells are at Tufts, Andrew, what is the trajectory of academic research using these acquired lines to make it more translatable to what industry values? Or David?
What is it? Okay, I think I understand the question, or at least I think I'm interpreting it in the right direction. So I think, well, I'll answer it in the way I think I should, and if you asked it and I answered wrong, just yell at me. So I think the trajectory, and maybe I'll just speak from like personal perspective, is I want to get out of well plates like stat and move into bioreactors and move into suspension systems, and I'm personally so scientifically excited by these cells because I just want to work in those systems that are more relevant to sort of the scale that I guess I sort of see our research heading in directionally. I think there's there's so many, and this isn't actually even sort of a suspension versus adherent thing, but there's so many mismatches between academic research and what companies are doing at production scale. And I'm just thinking about the fact that in academia, and maybe hopefully this isn't getting too into the weeds, but you change the media every 2 days, and you fully change the media every 2 days. And that's just not what happens in industry. More nutrients are added to a bioreactor. There's maybe this perfusion system. It's kind of, I always get so frustrated that we're kind of working in these ways that the ways that they're not translatable, and yet to date we've been stuck with them, and it feels like these cells and like working in this different type of system unlocks feeling that kind of mismatch. So I don't know if I answered the question at all. Positive trajectory, an exciting trajectory. Is that the question? Elliot, save me.
I'll just add that at least from our perspective, I think the impact of these cells is going to be dependent on how many people use them at the end of the day. And so, if we want to create like a quote unquote workhorse cell line for the field, then we need a lot of people using them. We need a lot of people publishing papers on it, so that that effect just sort of snowballs from there in a positive feedback loop. So one of the things that one of the projects that I'll hopefully lead this year is around sort of idea of flash grants that we can provide to people that have these cells, where they can perform specific sets of experiments to characterize some of these fundamental principles. If you look in the literature, in the CHO cell literature, the biopharma literature, a lot of those foundational experiments performed in the 80s and 90s enabled us to reach the scale and produce hundreds of billions of dollars worth of products that we produce today. And essentially, we really actually have to basically repeat a lot of that work for these cell lines in this field. And that's kind of step one. And so what we'll hopefully do is provide a little bit of an incentive in the form of small grants for people to do that sort of research, so that we can even accelerate the knowledge generation right from the get-go and push this cell line, as Andrew mentioned, that has the potential to be directly or more commercially relevant and translatable into the public knowledge.
Great. I think that's a good place to wrap up. Thank you. We have a break next, and we will have California cultured chocolate samples at the coffee station. So if you want to try cell cultured chocolate, head there. Thank you so much, and see you all at 3:45.